Hi there, this is Miss Pearson, and today I'm going to go over DNA replication with you. In another video, I'll go over the central dogma, but first we're going to jump into DNA replication. So we have to replicate DNA, which is the genetic material in every cell, because the DNA needs to be copied and then it can get divided into daughter cells. So if we just look at a little doodle here, if you've got your cell, and let's say we've got 2 copies of the DNA, well, in your daughter cells, then when they divide, each cell will have DNA in it. And DNA replication is important for cell growth and reproduction of organisms, along with repair. So step 1 in replication, we've got semi-conservative replication, so each strand has one parent strand and a new strand. So up here we've got this parent strand, we've got 2 yellow strands, so if you look down at the next step, you can see that there's the parent strand again, but this red one is the new strand. And same thing over here, we've got our parental strand and our new strand. And this parental strand, you can actually follow every time it gets replicated. So there's always going to be, in semi-conservative replication, one parent strand and one new strand. So here's a big picture of what that semi-conservative DNA replication looks like. All these different parts we're going to go through in these next slides. But here's just an overview of what that process looks like. So first, DNA helicase is going to unwind or unzip the DNA strand. So here, here we've got our DNA helicase working this way to break these hydrogen bonds, which keep our nucleotides together. So like our A's and T's, G's and C's, so it's breaking those apart. And these 2 strands open up to form the replication fork. All right. So DNA helicase unwinds or unzips the strand moving this way to break those hydrogen bonds so that the replication fork can form. After that, primase, it adds primers to create starting points. So it'll add this RNA primer here to each side of the DNA. And then that way DNA polymerase can come at this starting point and work its way towards the 5' end. So that DNA polymerase is adding nucleotides to replace those primers. So if we go back to this photo, that's what this is doing. This DNA polymerase is adding nucleotides here to replace these guys. So the new strand is built up in the 5' to 3' direction. DNA is anti-parallel. Both strands have opposite 5' to 3' orientations. So there's the leading strand, which is replicated as the DNA unwinds, and then the lagging strand is replicated away from the fork and creates these fragments, which are called Okazaki fragments, and then these fragments get connected later. So if we take a look at this picture at the replication fork, here's our leading strand, and down here is our lagging strand. So here we would have helicase breaking down those hydrogen bonds, breaking those apart, so we have our replication fork, and then we're going to have our new strand of DNA being made at the 3' end, but in a 5' to 3'. And then our Okazaki fragments, they also start at the 3' end, but they're going to be the opposite orientation. This will be 5', and this will be 3', and this will be 5', and this will be 3', 5', and 3'. And then later, these gaps here get joined. So these are, again, those Okazaki fragments. So after step 3, we have our 4th step where that ligase is coming in alongside and it seals the backbone. So ligase is what's sealing these gaps here in the Okazaki fragments. So it completes the joining of those, and then 2 new strands of DNA are done. And each one has a new half and an old half, right? So that's that semi-conservative replication. We have our parent strand. See if I can draw this for you. And then in our new DNA, we'll have one parent strand and then a new strand. Okay, so here's just a big picture of what we just kind of walked through, right? So we've got our DNA and our helicase is coming, and it's breaking those hydrogen bonds so that we have our big replication fork here. And then we're going to have that primer get laid, and DNA polymerase will start there so that it can start laying nucleotides in the correct direction. And it'll do that for both the leading strand and the lagging strand. So here's our lagging strand working this way, but in different fragments, those Okazaki fragments. So a review of replication, we have DNA helicase unwinding that DNA. Our RNA primers will bind to the DNA strands, and then DNA polymerase is going to add nucleotides to the DNA. So then we have our leading strand, which is the continuous adding of bases, and our lagging strand, which are the Okazaki fragments. And then DNA ligase comes and fills in those gaps. Okay, so this is a great video to review DNA replication, which I will, you'll have available for you on Google Classroom. But next we're just going to talk quickly about prokaryote and eukaryote replication. So in prokaryotes, there's only one origin, as you can see in this picture. But in eukaryotes, there can be many origins. We've got 1, 2, 3 origins where DNA replication is actually taking place. So if you look at RNA, which is ribonucleic acid, so its name comes from RNA, ribonucleic acid. Why do cells use RNA? Well, the DNA can't leave the nucleus. So a copy of that DNA can actually go into the cytoplasm and guide the synthesis of proteins. So we'll talk about that later in the central dogma video. But for now, little comparison here then. We've got DNA with 2 strands, and RNA is a single strand. And then the type of sugar that DNA uses is deoxyribose sugar, and then RNA uses ribose sugar. And the base pairs for DNA are A, T, C, G, and RNA is A, U, C, G. So in RNA, those T's actually will be U's. Okay, that's all I have for DNA replication. But the next video will be the central dogma, so be sure to watch that one as well.